The new version of PREDetector proposes two types of graphical outputs at the top of the page. The lower part of the screen is dedicated to the table containing each hit, ranked according to their score. The first panel, entitled Plot, contains a gray bean plot that displays the total number of hits according to the score. The yellow circles represent the scores of the sequences used to generate the matrix. The red line provides a mean for users to estimate the reliability of the predicted hits. Indeed, this line displays the proportion of hits that localize in the intergenic versus the coding regions at a given score. By default, the threshold score is fixed to TMS, represented by the dotted red line, which is the threshold imposed by the minimal score of sequences used to generate the position weight matrix. This plot is interactive. Users can adjust the threshold score as they wish, with immediate adaptation in the genomic context graphs displayed in the right panel, but also in the output list in the table that ranks the predicted hits according to their score. The second panel, called Genomic Context, displays the distribution of the predicted binding sites on the targeted genome sequence. Two lines representing the genome are shown. The bottom line displays the full genome sequence, with orange bars representing the coding sequences and the blue bars the position of the identified DNA motifs. This part of the graph is also interactive, as users can select a zone of the genome to be more accurately visualized, and that will be displayed in the line above. Placing the cursor on an orange bar will give you the predicted function of the corresponding gene. The table below the graphs lists all the regular prediction hits. This table is also interactive, since you can order the results by clicking on the up-down arrows in any columns or even search for text. You can also click on any row to update the genomic context and zoom on the selected transcription factor binding site. Clicking on the downwards arrow icon will display the output list by grouping hits that belong to the same intergenic region. Clicking on the floppy disk icon will allow you to save the current results obtained with the chosen threshold score and your own modifications. You can access your work in the Regulon Prediction tab. The first part of the PDF report summarizes the input information and the selected screening parameters. The second part shows the graphs associated with the prediction. And the third part of the report displays a table that lists the putative binding sites and the associated genes predicted to be controlled by the studied transcription factor.